Dear students, welcome to all of you. Today we are here to discuss cytology in relation to taxonomy or cytotaxonomy. First, the introduction. Cytology refers to the study of structure and function of cells, and cytotaxonomy is the utilization of cytological characters for solving taxonomic problems. However, mostly the information about the chromosomes, that is, number, shape, and size of chromosomes is taken into account. Thus, systematic application of chromosomal information may be called cytotaxonomy. Taxonomy and cytology study the same phenomena but from two very different angles. Cytology, or more properly karyology, concerns itself with the architecture of the germplasm, taxonomy with the adult forms which result from germplasm. Cytology has made an outstanding contribution to taxonomy during the past few decades. Since the cytological data are directly derived from the nucleus, the seat of hereditary material, they may be used for understanding the evolution and relationships of populations. The chromosome studies serve as a source of data for biologists concerned with systematic and evolutionary investigations. The characteristics of the chromosomes which have proved to be of taxonomic value include chromosome number, chromosome structure, and chromosome behavior at meiosis. First, the chromosome number. Chromosome number is an important taxonomic character as it is probably one of the most constant single feature employed. All individuals within a genus usually have the same chromosome number. For example, all known species of Platanus have X is equal to 7 basic chromosome number. All species of Quercus have N is equal to 12 haploid chromosome number. But there are exceptions also. For example, in Moringa, the haploid chromosome number may be N is equal to 11 and 14. Chromosome numbers recorded for spermatophytes vary from 2N is equal to 4 in Haplopappus gracilis of Astraceae to 2N is equal to 500 in Kalanko species of Crassulaceae. Some pteridophytes, like the members of the family Ophioglossacea and Polopodiaceae, show unusually high chromosome number. The highest chromosome number has so far been recorded in Ophioglossum reticulatum, that is 2N is equal to 1260. In general, the number of chromosomes in each cell of all the individuals of a single species is constant. The more closely related species are likely to have the same chromosome number, and the more distantly related are likely to have different numbers. This relative conservativeness renders chromosomes an important and much used taxonomic character, and is in fact just about the only biosystematic evidence which is consistently recorded in standard floras and the like. Usually, the information is provided in the form of the diploid number, that is 2N. In reporting new chromosome counts, it is usual to quote the diploid number 2N when the count is based on mitosis in sporophytic tissue, and the haploid number N when it is based on mitosis in gametophytic material or meiosis. In somatic tissue, haploid or diploid counts are usually made on rapidly and synchronously dividing cells, for example, meristems, embryos, and young sporogenous tissue. The suffix ploid refers to the chromosome status. The prefix indicates the degree of the ploidy. When various members of a taxon possess an exact multiple of the basic number of the chromosomes, the series is called as polyploid. Polyploidy is widespread in plants. About 50% to 70% of angiosperms so far investigated are reported to be polyploids. For example, in the genus Salix, there are species with 2n is equal to 38 Salix viminalis, 2n is equal to 76 Salix atrocinerea, 2n is equal to 114 Salix falcifolia, and 2n is equal to 152 Salix myrsinitis. These numbers are based upon 19, the gametophytic chromosome number of the diploid species. Similarly, in the genus Festuca, there are species with 2n is equal to 14, 28, 42, 56 and 70, the base number being 7. Another interesting example of polyploid series is found in the genus Pteraxcum with 2n is equal to 16, 24, 32, 40 and 48. This type of polyploidy is known as euploidy because the number is an exact multiple of the haploid number, that is n is equal to 8. Polyploidy is known to arise by either somatic or meiotic processes. In the former case, the chromosome number of a cell becomes doubled by mitotic chromosome division not being accompanied by cell division. This cell, if it is part of a young embryo or meristem, can soon develop into a substantial piece of tissue which gives rise to a plant or shoot which produces flowers with diploid microspores and megaspores and hence tetraploid seeds. In the latter case, a diploid plant produces diploid microspores and megaspores owing to pre-meiotic doubling or to non-reduction at meiosis, tetraploid or triploid seeds again being the result. If the diploid plant referred to in these two methods are distinct species whose genomes can be represented as AA, then the triploids derived from them will be autopolyploids or autoploids designated as AAAA. But if the diploid plant was a hybrid between two plants with unlike genomes, that is AB, then the derived triploid will be an allopolyploid or alloploid designated as AABB. In the case of allopolyploids, the genomes of the parental diploids may have the same base number, that is monobasic polyploidy. For example, in Festuca, or the polyploid may incorporate genomes of different base numbers, that is dibasic polyploidy. For example, the three species Spartina maritima with 2n60, Spartina alterniflora with 2n62 and Spartina angelica with 2n122. In such cases, the derived allopolyploid possesses a new base number. In Spartina, the new base number x is equal to 61 has originated from parents with base numbers of x is equal to 30 and x is equal to 31. Quite often, a polyploid complex consists of a series of distinct diploid species which have hybridized and become polyploid to produce a range of tetraploid, hexaploid and sometimes higher levels of ploidy. Because of the recombination of genomes, the distinction between the taxa which exists at the diploid level often becomes blurred at the tetraploid and higher levels, and distinct taxa cannot be recognized. Such a situation is described as polyploid pillar complex, and examples are to be seen in the Dactylus glomerata, Juncus bufonius complexes. The names Dactylus glomerata and Juncus bufonius strictly refers to the tetraploid and hexaploid, octoploid representation of their complexes respectively. In both cases, a number of diploid species can be segregated from them. The type of polyploidy referred to hitherto is known as euploidy, but aneuploidy, where chromosome number vary not in multiples of the basic set but in single or few chromosomes only, is also common. In the genus Vicia, there are chromosome numbers of 2n is equal to 10, 14, 24 and 28, obviously consisting of aneuploids clustered around the diploid and tetraploid levels. And in Crepus, 2n is equal to 6, 8, 10, 12, 14, 16, 18, 22, 24, 42, 44, 66, 88 and some other numbers with less obvious clustering. Where the numbers bear no obvious relation to a particular series of ploidy levels, the term dysploidy may be used. Now the chromosome structure. In addition to variation in number, chromosomes vary in form, size, volume and in the amount of distribution of heterochromatin. In most plants, chromosome length varies from 0.5 to 30 microns. The monocotyledons usually have larger chromosomes than the dicotyledons. Generally, woody plants have smaller chromosomes than the herbaceous ones. The location of the centromere determines the relative length of chromosome arms. The position of centromere varies from chromosome to chromosome and it provides following different shapes to the chromosomes: telocentric, acrocentric, submetacentric and metacentric. Telocentric. When the centromere is at the terminal or proximal position, the chromosome is called telocentric. The chromosomes are rod-shaped. Telocentric chromosomes are very rare. In telocentric chromosomes, one arm is very long and the other one is very short. The short chromosome arm is designated P, that is petite, and the long arm Q, one letter after P. Acrocentric. When the centromere is situated near one end, forming a long arm and a very short arm or even imperceptible short arm, and the chromosome appears rod-shaped. Submetacentric. When the centromere is situated slightly away from the middle and one arm of the chromosome will be shorter than the other. Such a chromosome will appear L-shaped during anaphasic movement. Metacentric. When the centromere lies at or near the middle of the chromosome and the two arms of the chromosome are almost equal, then it is called as metacentric. During anaphasic movements, the chromosomes bend at the centromere so that the metacentric chromosomes are V-shaped. The V types of chromosomes have two equal arms and a median centromere and are termed as symmetrical. The other types are called asymmetrical. In addition to the size and position of the centromere, the third useful aspect of chromosomal gross morphology is the position of secondary construction. The presence of satellites is important not only for their intrinsic taxonomic value, but also because they can be confused with additional chromosomes. The appearance of the basic chromosome set under the light microscope is known as the karyotype. For example, the karyotype of Calistegia 2n is equal to 22 as consisting of 8 short metacentricus, 2 short submetacentricus and 1 short metacentric with an addition a well-defined secondary construction and satellite. For more accurate purposes, arm ratios can be used. Karyotypes are usually calculated from the means of a substantial number of observations and often they are represented diagrammatically as ideograms or karyograms. These aspects of chromosome structure together with chromosome size and number, have been found extremely useful at all levels of the taxonomic hierarchy. Karyotypic study of aconitum reveals that there is variation in chromosome length and the position of centromere from species to species. Most specialized karyotype is seen in the most highly and recently evolved animal species in the tribe Helibori of family Ranunculaceae. Most asymmetric karyotypes are reported from Delphinium and Aconitum, the two most advanced genera. In the Luzula spicata group, for example, plants can have 2n is equal to 12, 14 or 24, but the total chromosome volume is about the same in all and indicates that the higher members are probably derived by fragmentation, that is agametoploidy. In other cases, different chromosome numbers occur in different cells of one root tip, that is mixoploidy. Sex chromosomes are rarely of much taxonomic value in plants, as diocism, at least in higher plants, is relatively rare, and even when it does occur, as well as in most lower plants, sex chromosomes are often more discernible. However, Silene dioica has an XX XY system, Rumex acetosa has an XX XXY system, and in many liverworts there is an XX XY system. Now, the behavior of chromosomes at meiosis. The study of chromosome pairing is one of the major means of investigation in cytogenetics, the study of the role of the chromosomes in heredity. Some taxonomic information can be gained from the study of the mechanisms of meiosis itself. For example, meiosis in some Zoncaceae and Cyperaceae, which have small chromosomes with unlocalized centromeres, is considered to be inverted. That is, equational division precedes the reductional division. Many peculiarities of meiotic behavior owe their origin to the existence of heterozygosity, so that at meiosis one is observing the pairing of unlike genomes. The differences which can exist have often arisen by duplications, deficiencies, inversions or translocations of chromosomal material, and the meiotic configurations usually provide evidence of the precise nature of these rearrangements. Some species are known to be consistently heterozygous for certain translocations, which give rise to multivalent formation at meiosis. In the genus Oenothera, all species are diploid with 2n is equal to 14. Many of these exhibit a normal meiosis, but in the subgenus Oenothera, the species are heterozygous for translocations involving varying number of chromosomes. A study of chromosome behavior during meiosis can help interpret aneuploidy and polyploid changes and also identify other structural modifications that are commonly involved in chromosomal evolution, for example, deletion, duplications, inversions and translocations. Dear students, now we will discuss the use of cytological variation at family level. Chromosome numbers do not have much value at higher levels like family level or above from classification point of view. In the family Ranunculaceae, chromosome number and chromosome morphology have provided basis for more natural arrangement of genera and tribes. Engler and Prentl have recognized two major tribes, Helibori and Anemone, in the family Ranunculaceae with base chromosome number of 7, 8 and 9, and both have the genera with large and small chromosomes. The genera Aquilegia and Thalictrum, along with a few others, differ from most Ranunculaceae in having small chromosomes and basic chromosome number being 7 rather than 8. These genera have thus been segregated into a separate tribe Thalictri. Two other genera of the Ranunculaceae, Coptis and Xanthorrhiza, with small chromosomes and the base number 8, have been removed to an additional tribe, Coptidae. The genera Agave and Yucca placed two separate families, Amaryllidaceae with inferior ovary and Liliaceae with superior ovary respectively, by some earlier workers. Due to the overall similarity in these genera, they are now generally separated from their respective families and placed in the Agaveaceae. This classification is supported by cytological evidence, that is, Agaveaceae are characterized by very distinct bimodal karyotype comprising 25 small and 5 large chromosomes. Their karyotype similarity justifies their inclusion to a single family Agaveaceae, as has been done by Hutchinson. The genus Biberstinia was earlier included in the family Gyraneaceae. Takhtajan, 1987, recognized a separate family Biberstiniaceae for the genus Biberstinia on the basis of presence of endosperm and basic chromosome number X is equal to 5. In Gyraneaceae, the basic chromosome number is X is equal to 7 to 14. The major subdivisions of the family Poaceae, that is, subfamilies, tribes and genera, as recognized currently, are characterized by the number and size of the chromosomes. For example, subfamily Poidae has basic chromosome number 7, but certain tribes, for example Glycerae, X is equal to 10, or genera Anthoxanthium, X is equal to 5, within it deviate consistently and illustrate the relative nature of the concept of base number. The base number of various subfamilies of Rosaceae is 7, 8 or 9, but the subfamily Pomoidae has 17 as base number. This suggests that the members of subfamily Pomoidae are either polyploid hybrids between taxa with X is equal to 8 and X is equal to 9, or are polyploids of taxa with X is equal to 9 with loss of one chromosome. Now the cytological variation at and below genetic level. There are several examples where chromosome numbers support genetic status. 3 species of Chlorophytum, that is Chlorophytum barochi, Chlorophytum glaucum and Chlorophytum glaucoides, are very similar to each other morphologically, and therefore it is difficult to identify them easily. Cytological investigations revealed that Chlorophytum barochi has 2n16 and the other 2 species have 2n42. The 2 species with 2n42 differ in their karyomorphology. The genus Cistus of Cistaceae was earlier included in Helianthemum. The former has base number 8 and the later 9. This supports the recognition of Cistus as a separate genus. The 2 species Cycindia filiformis and Microcala pusilla of the family Gentianaceae were previously included in the same genus Cycindia. Their base chromosome numbers are X is equal to 13 and X is equal to 10 respectively, and this supports their separation into 2 distinct genera. Chromosome number is of little value if both aneuploidy and polyploidy occur in the genus, as in Crepus. However, if the chromosome number is constant within a genus, this is particularly useful in generic delimitation. In the genus Tephrosia of Fabaceae, all the species possess 2n22 except Tephrosia constricta with 2n is equal to 16. This species has been treated as a separate genus Sphyctospermum, and the chromosomal studies supports this separation. Many workers have recognized the genera Physaria and Lasquirella of the family Brassicaceae as a single genus. Cytological evidence suggests that these two genera should remain separated. Artemisia is the largest genus of the tribe Anthemidae and one of the largest of the Asteraceae. It contains more than 500 taxa. Artemisia has two basic chromosome numbers with ploidy levels ranging from diploid to dodecaploid for x is equal to 9 and from diploid to hexaploid for x is equal to 8. Chromosome number in diploid is most often 2n is equal to 18 or 16. The highest chromosome number known is 144 and aneuploidy is frequent. Within several species, different ploidy levels have been observed that were considered to rest on autopolyploidy. However, based on characters such as intermediate morphology and geographical distribution, Ehrendorfer 1964 suggested that several Eurasian polyploids were in fact allopolyploids. Although much cytological work has been done in Artemisia, definite evidence for allopolyploid is scarce. The karyotype data have been extremely useful in distinguishing various species of Clarkia, Viola, Nicotiana, Potentilla, Achillea, Gossypium, and many more genera. The cytology of 13 taxa and 2 hybrids in the genus Pennisetum indicated the distribution of the taxa among the 4 basic chromosome numbers 5, 7, 8 and 9. The diploid nature of Pennisetum ramosum and Pennisetum typhoidis and the genomic allotetraploid status of Pennisetum purpureum was further confirmed. Pennisetum messicum with 2n is equal to 32 and Pennisetum oriental with 2n is equal to 36 and Pennisetum subangustum with 2n is equal to 36 suggested probable autotetraploid nature, and the 3 hexaploids investigated, that is Pennisetum polystachyon, Pennisetum longistylus and Pennisetum sequamulatum with 2n54 revealed allohexaploid constitution. The small section capillaris of the genus Caraccas has been studied from the cytological and taxonomical points of view. It comprises 9 distinct species. 3 species include 2 distinct subspecies each. 2 varieties and 3 formae are accepted for 2 species respectively. The chromosome numbers of the taxa involved are 2n 18, 36, 38, 40, 54, 56 and 58. It is pointed out that the chromosomes of Caraccas have a polycentric or diffuse kinetochore, and that this explains the aneuploid variations in number known to be typical of the genus. Karyotypic analysis of the chromosome complements of the section capillaris revealed that in the basic diploid set of 2n18 chromosomes, 4 units are long, 10 are medium, and about half the length of the long ones, while 4 are small and about half the length of the medium ones. This frequency of size class is also met within the plants with 2n is equal to 36 and 54 chromosomes. Those taxa with 2n, 38, 40, 56 and 58 chromosomes show, however, such a deviation in the frequency of the size classes that it seems logical that these numbers have been produced by a secondary fragmentation of the chromosomes. On the basis of karyotypic analysis of this section and other groups of the genus, it is concluded that the primary basic number for the genus Caraccas in particular and the family Cyperaceae in general must have been x is equal to 5, all other numbers having been derived mainly by fragmentation of chromosomes with a polycentric or diffuse kinetochore. This phenomenon of partial agametoploidy is widespread in Cyperaceae, whereas complete agametoploidy is typical of the closely related Juncaceae. From the above discussion, we can conclude that chromosome studies serve as a source of data for biologists concerned with systematic and evolutionary investigations. Dear students, this is all about cytotaxonomy. Dear students, we have discussed over here various characteristics of chromosomes which have proved to be of taxonomic value. We have also discussed the use of cytological variations at family level and at and below genetic level. Thank you very much. God bless you.